Well, welcome everyone to our first community chat series. Today we are meeting with Dr. Steve Baker and his research team at University of Utah at our Creatine Deficiency Research Center. Dr. Baker, as most of you probably know, is leading an exciting project at the Research Center to treat CTD with an innovative and new approach to gene therapy. The CDRC was launched earlier this year through funding raised by ACD in last year's Holiday Heroes fundraising campaign. And Sangeetha and I, who is also joining our call, actually got to go visit the team last week during the consensus workshop that was in Salt Lake City. And we got to see some of the work that you're going to hear about today. So with that, I will turn it over to Sangeetha.
Thanks, Heidi. Yeah, it was a really fun visit. Couple weeks ago, we got to see patient fibroblasts growing in these super large culture plates. We got to see iPSCs that were soon to be differentiated, and we saw the confocal microscope where all of the magnificent pictures and slides that Steve and his team prepare are actually taken. It was great, and these fibroblasts and iPSCs have been instrumental thus far in the research plan itself, and we're going to talk about that very soon. But before I hand it to Steve and his team to walk us through what they've been up to these past several months, maybe a brief refresher of the research proposal is in order. So I'm going to summarize over here, Steve, what I understand, and I'll leave it to you and your team to walk us through the specifics. But just as a high level overview, your proposal focuses on testing the hypothesis that AGAT and GAMT enzyme can be used to get cells to make their own creatine. So even when these cells don't have AGAT naturally, you can introduce AGAT enzyme exogenously and then coax the cells to make their creatine. And this should work as a treatment for CTD, because then even though the cells don't have a functional transporter that can bring in creatine, they should not need this anymore, because they'll be able to synthesize or make their own endogenous creatine because of these exogenously added enzymes. And as such, this type of treatment would be applicable to any type of mutation bearing cells. So, or more specifically, any type of CTD mutations. So applicable really to the whole gamut of the CTD patient community. That's the high level bit, but I'll hand it over to you, Steve, to just tell us a little bit more about the specifics, and then share with us some exciting new data.
Yeah, sure. Thank you so much, first of all, Heidi and Sangeetha, for hosting us. And it's been such a great partnership with ACD and the families and patients and the research community. And we couldn't be more proud to be a part of this community. And I'm here today with my awesome team that has really produced the data that we'll share with you today, and we'll talk about the concepts behind that strategy and why we think it might be a promising strategy for treating CTD. And so I'm really honored to have John and Chloe here as well to help me kind of explain what the lab is doing and the progress that we're making. So let me share my screen here, and I will, just as a kind of a background for what we're working on, go over some old data and then maybe some newer data. So the kind of the foundation for this, the work that we're doing, came out of work that I had done as a postdoctoral fellow. And so around that time, the kind of dogma in the field was that creatine is synthesized. So we know that your body synthesizes about half of its daily needs for creatine on a typical Western diet. The other half, of course, coming from intake of things like protein and meat and fish. And so the creatine that is made by our own bodies was thought for a very, very long time to come from the cooperation of two organs, which is essentially the kidney and the liver. And early studies going back decades were able to identify the enzyme activity that is found in the kidney. And that enzyme is called AGAT. So AGAT is the first enzyme in the synthesis pathway, and it takes two amino acids, glycine and arginine, and it literally stitches them together to make an intermediate called guanidino acetate. That guanidino acetate was thought to be transported through the blood, taken up by the liver, and then worked on by a second enzyme called GAMT. And that GAMT enzyme was known to be very high inside the liver. And it takes guanidino acetate. And all it does is take a tiny little methyl group, a little single carbon, and add that single carbon onto guanidino acetate. And that makes creatine. And then it was thought that the liver then releases this creatine into the blood. And then when it's inside the blood, it could be taken up via the creatine transporter into various cell types that need it. For example, the brain, skeletal muscle, and heart. And so this was the kind of prevailing view of creatine metabolism, even in this published review in 2020. But when I was a postdoctoral fellow, what I did was I used a bunch of antibodies. So I screened a large pool of antibodies to find ones that were very specific and could be used for immunofluorescence. So a technique where we take a tissue and we label where certain proteins are in a tissue by making that protein glow. And we do it in a very specific way such that that protein glows only in exactly the cell and exactly the subcellular place where that protein is. And so here's just some of these glowing images where I stained a section of mouse liver for either AGAT, which is the first synthetic enzyme, or GAMT, which is the second synthetic enzyme. And where those enzymes are, the tissue glows red. And so, of course, we weren't surprised to find that the liver had a lot of GAMT, because the prevailing dogma in the field was that GAMT is really, really high in the liver, and that was the function of the liver in creatine synthesis. But we were surprised to find that there was AGAT signal coming from the liver. And more importantly, it wasn't coming from the majority of the cells inside the liver. So your liver is comprised primarily of a special cell type called hepatocytes. And those hepatocytes synthesize all the protein in your blood, or the vast majority of proteins in your blood, to keep the blood coagulation system and hormone transport system functioning. And so those guys, one of the proteins that we can use to identify hepatocytes is a blood protein called albumin. So a large component of the protein in your blood is albumin, and it's specifically synthesized by hepatocytes. And so we find those hepatocytes by labeling for albumin. And what we found was that the AGAT signal actually wasn't overlapping with that albumin signal, but instead it was coming from a different cell type, which is marked by a different antibody. Here we're just calling it endothelium. But the endothelium are the type of cells that line the vasculature. So all throughout your body, blood is pumped by the heart, and it perfuses every single organ. And the vessels that it travels through are lined by a specialized cell type called the endothelium. And so what we were seeing was that AGAT overlaps with the endothelium, but not with the hepatocytes, which instead GAMT was overlapping with the hepatocytes. And just to impress upon you kind of how this looks in 3 dimensions. So this is a confocal microscope image where we're literally, as we're traveling through the liver, we're going section by section by section of a block of liver tissue. And here we've stained it for albumin, so that green marker, which is labeling the hepatocytes. And then the glowing little red dots, you can see in 3 dimensions that they overlap with these like little black empty parts of the tissue. And those black empty parts are actually the blood vessels. And they're lined by those endothelial cells, which we highlight with this purple marker where we overlap both of those images together. And so again, this convinced us that the liver expresses both AGAT and GAMT, and not just GAMT only. And so to sort of delve into this a little bit more, we turned to a newer resource, which had been published in 2018, where they took 4 male mice and 3 female mice, and they separated all of their cells from all over the body, and then quantified how much of each gene is expressed in each cell type. And so when we looked at this large data set that was publicly available, of course, we weren't surprised to find that GAMT is really, really high. So the y-axis here is GAMT, and the x-axis is AGAT, which its gene name for AGAT is GATM. And so we weren't surprised to find that hepatocytes had a lot of GAMT inside their nucleus and cytoplasm that they were expressing. And consistent with our immunofluorescence data, we found that the blue circle here is the liver. The endothelial cells in blue actually did express a lot of AGAT and not GAMT, which is exactly what we were seeing. But when we made this plot, something pretty obvious jumped out to us. So I'm not a very smart person, but I did notice that there was this big green circle in the top right. And this big green circle is the oligodendrocyte. And so what we were surprised is that oligodendrocytes, which are this specialized cell type inside the brain, expresses more GAMT than hepatocytes. And it actually expresses more AGAT than kidney cells. So these are the cells in the kidney that express AGAT, which is the proximal convoluted tubule cells. And so these oligodendrocytes seem to be like these little factories because they produce both AGAT and GAMT, and they're living inside of your brain, expressing both of these enzymes at the same time. And so we wanted to see if this was true. So this was true at the gene expression level when we measure what's called the RNA. So the RNA ultimately is what makes the protein down the line. But we wanted to confirm that truly the protein was there. And so again, we turned to immunofluorescence where we made the proteins glow inside of a mouse's brain. And when we did that, here you're seeing a section of mouse cerebellum. And the neurons are highlighted in green here. And you can see that the red dots, which are the AGAT signal, is not coming from the neurons, but it's coming from these cells that are all inside what is called the white matter, which is chocked full of oligodendrocytes. And when we quantified this across a couple of different parts of the brain, we found that oligodendrocytes indeed expressed the vast majority of AGAT across the brain. And we wanted to do the same thing with GAMT. And when we stained for GAMT, again, these neurons are here shown in green. They did have a little bit of GAMT expression, but really a ton of GAMT expression was coming from these cells in the middle of where the neurons aren't, which are oligodendrocytes. And again, we looked across the brain, and here I'm just showing you in the cortex, you do have these cells that are positive for this marker called NuN, which stains neurons, but right next door to them are these oligodendrocytes that are also expressing GAMT. And so it seems that inside your brain, AGAT is somewhat specialized for expression in oligodendrocytes, although we did see some minor expression in other cell types. But GAMT was also expressed in oligodendrocytes, consistent with the RNA-seq data, and it was at very high levels for both. Whereas neurons, it seemed to only express GAMT. And so we wanted to see, was this true in the human brain? So everything that I've shown you up until now has been mouse tissue. And so we turned to a technique called ATAC-seq, and another postdoc in the laboratory that I did my postdoc in was working on ATAC-seq inside the human brain. And ATAC-seq, suffice it to say, is a specialized technology where we can see whether genes are sort of on or off by looking at the DNA around them. And without getting too far into the weeds, suffice it to say that in oligodendrocytes and in oligodendrocyte precursors, we saw a very nice big peak right over the AGAT gene, which is called GATM. And then also in oligodendrocytes, we saw these nice peaks over the GAMT gene. And so, but we did see a little bit of these peaks inside neurons. And so that suggested to us that our immunofluorescence data from the mouse brain was probably also reflecting what's going on inside the human brain. And I think that's important if you're going to design a gene therapy approach that relies on those mouse data. And so we turned to the literature given this information, and we noticed a couple of key observations. So I'll just summarize kind of what was known at the time, which is that in mammals, the brain tissue creatine concentration is very, very high. So the salts that are inside your blood are in the range of millimolar. And those are very, very high concentrations. Most of the medications that we take orally usually achieve tissue concentrations in the nanomolar to even low micromolar range. So to have something in millimolar which is like 1,000 times more than a micromolar, and 1,000,000 times more than a nanomolar, is something that's very, very high. And so the creatine is just like that. So creatine is enormously high inside the tissue, so in the 10 to 20 millimolar range inside the brain. And this those measurements of direct measurement inside mammalian brains are confirmed with estimates from human MRS, or magnetic resonance spectroscopy. And so even though creatine is really, really high in the brain, it's at much lower concentration inside the CSF. So the CSF is the cerebral spinal fluid. It's the fluid that bathes the brain, and all the neurons are suspended in this extracellular fluid, this CSF. And the creatine concentration inside the CSF is in the micromolar range. So really like 1,000 times less concentrated than the amount that we see inside the brain. And this suggests to us that the vast majority of creatine is probably intracellular, right? Because if the extracellular concentration is 1,000 times less than the total tissue concentration, it's the other remaining 999%, right, of that 999 of that 1,000-fold have to be coming from the intracellular compartment. And so what's interesting is that in patients who have CTD, the CSF creatine concentration is not decreased. It's actually normal to the average is slightly elevated. Yet we know that in patients with CTD, if you use MRS spectroscopy to measure how much creatine is inside their brain, it's about 20% of a healthy normal brain. And so unfortunately for us, the resolution of MRS spectroscopy is only about 10 millimeters in width. So we can't tell where is this deficit coming from, but our strong suspicion is that the deficit that we see by MRS spectroscopy in patients who have CTD is coming from the intracellular compartment. But the thing that we're not sure of is what cell type it is, but we strongly suspect that it is neurons. And so our model is that creatine inside your brain is coming from a very specialized cell type, so those oligodendrocytes. Those oligodendrocytes are actually known to be like nurse cells for neurons, and they hang out right next to the neuron axon, and they do a special job called myelination. The details are not super important, but suffice it to say they're professional helper cells for the neurons. And so one of the jobs that we think they're doing is that they express both AGAT and GANT in order to synthesize creatine, and then they feed that creatine to neurons, and neurons rely on the transporter that's lost in patients with CTD. So that CT1 transporter, we know by RNA expression, is present on neurons. And we think that those neurons are using CT1 to take in creatine into their cytoplasm, where it can then be worked on by creatine kinases. And in particular, there's one creatine kinase called CKMT1 that is almost exclusively expressed inside neurons. And that makes us suspicious, because in order for creatine to work, it has to have the creatine kinase to do its thing. And so we strongly suspect that it's the neurons that need that creatine. And so what we know from our immunofluorescence data and the RNA-seq is that actually neurons express GAMT, but they don't have that first synthetic enzyme AGAT. And so if they have GAMT and they need creatine, our sort of model, or our therapeutic approach, is that maybe we can get them to express AGAT so that we can complete the circuit within the neuron, so that they're not reliant on this helper cell, which expresses both enzymes. Instead, make the neuron express both enzymes, and then you don't need that transporter to get it across the cytoplasmic membrane of a neuron, and the neuron can make its own creatine. And so essentially that is our gene therapy approach that we're trying, is that we hypothesize that patients with CTD have a decreased intracellular concentration of creatine due to a failure of neurons to uptake creatine from their neighboring oligodendrocytes. And so what we propose to do is to express AGAT inside of those neurons in order to complete the circuit. And if that means also that we overexpress AGAT and GAMT to ramp up the production, we're also amenable to that approach as well. And so in order to achieve this task, what we're planning to do is we're planning to carry out these experiments in mouse neurons, but then we'll share with you a little bit of what we're doing and why it's so important that folks in the research and the family and patient community get together to collect a repository of fibroblasts and other cell types that we can use as model systems to test our approach and for other labs to test their approaches as well. And so we're going to use mice. And when we wrote the initial proposal, this is still our plan, but in the meantime, we're also using patient primary cells. And so the idea is to take a wild type and a SLC6A8 deficient mouse, right? So SLC6A8 is the gene that's mutated that is causing the loss of that CT1 transporter in patients with CTD. And so the idea is to harvest neurons from either the wild type mouse or the SLC6A8 knockout mouse, and then deliver AGAT by lentivirus in order to complete that circuit where they have their own natural GAMT and then also would deliver AGAT. And then at the same time, as a parallel approach, we're trying to deliver both enzymes to see if that actually can ramp up production. And so as we're getting to these experiments, so in order to perform these experiments, we have to have ethics approval, we have to have mouse space. And so we just recently got our what's called IACUC protocol approved. So that's a huge step in the direction of doing these experiments. So that was just approved last week. And so in the meantime, we've been working on primary patient samples in order to see some preliminary data in this vein. And so let me just share with you some of the preliminary data we have. And so, sorry, cut me off if you want to redirect Sangeeta or Heidi. But is this okay if I just proceed?
This is great. Yeah. Keep going.
Okay, so I'm so happy to show you some of the updates that we have from last year. And again, this is all of the hard work of John and Chloe, and they've been doing phenomenal experiments in the lab and making really, really great progress. So the first thing that we did was we cloned the human AGAT gene and the human GAMP gene into a lentiviral vector in order to produce virus. So we delivered that to what's called HEC293 T cells. And so these are a common human model system. So they're human cells, but they're derived from a kidney. But they're very commonly used in the research community to test gene expression of different genes in a human cell line. And so when we deliver an empty vector, that's a vector that has no AGAT or GAMP there. And so what you can see is that HEC293 T cells express a little bit of their own GAMT, but they don't really express any AGAT. And so this is what's called a Western blot. A Western blot is just looking at, hey, can I see this protein coming from the inside of the cell? So what we do is we take those cells, we lyse them using a detergent, and then we run them out on a gel and detect the proteins using an antibody. And so when we do this, we can overexpress human AGAT inside these HEC cells, and they express a lot of it. And when we overexpress GAMT, we see a lot of that, especially relative to the natural amount that is in HEC293 T cells. And then what we were able to do in order to distinguish the overexpressed protein from the endogenous copy is we can add a tiny little protein tag called a V5 tag. Suffice it to say, it's just a little tiny piece of protein, which we hope doesn't affect the activity of the enzyme. And we can express AGAT with that V5 tag. We can express GAMT with that V5 tag and detect them as well. And we can do both together. So we can express AGAT plus GAMT in the same cells. And the cells seem relatively happy. They're not detrimentally affected by this, which is, I think, whenever you're planning a gene therapy approach, it's very important to be cautious and to have to have guarded expectations that what we're going to do here is going to lead to benefit, and we need to make sure that we don't cause deleterious outcomes. And so that's one of the things that we are very aware of, and we are with each additional step as we proceed, we're proceeding with caution and guarded optimism. So we want to see if this is going to work, but we have to make sure that it's safe before we would ever try this inside of a patient. And so we did this then with patient fibroblasts. And again, these were donated by members of the community. And and I'm just going to minimize my little screen here. There we go. Okay, in patient fibroblasts, so we had a line of fibroblast from a CTD patient with a very large deletion that would lead to no production of CT1. And so in essence, these are null fibroblasts, so they don't have a functioning CT1 transporter. And in those, you can see that a lot like HEC cells and a lot like neurons, there's just a tiny little bit of GAMT expression, but we didn't really detect any endogenous AGAT expression, which is, it's a very common cellular phenotype that we're noticing. But we can overexpress AGAT, we can overexpress GAMT, and we can do them both together with the tag and without that V5 tag. And the fibroblasts seem to handle this well, and they can really ramp up the production of AGAT and GAMT protein. And so then we wanted to see what does this do for creatine production? And so the first place we turned was HEC293 T cells when we overexpressed AGAT and GAMT in those. And I'm just going to let John. So John performed the previous experiment and then also this experiment. I'll let John kind of describe what he's found.
Can I ask just a clarifying question?
Yeah.
So you explained that, the first column of results were an empty viral vector, and then you used the terminology like we overexpressed. Are you saying that you delivered a different virus that did have a payload in it, and that's what you mean by overexpressed?
That's a perfect question. Exactly right. So yeah, so the empty viral vector, all it does is confer a antibiotic resistance gene. So it doesn't deliver any payload other than essentially a dummy gene that we can use to select for whether or not they got infected. But when we actually have our payload vectors, those are loaded with the gene that encodes human AGAT, or they're loaded with the gene that encodes human GAMT. And so we are delivering those genes via a lentivirus, which is a common strategy to deliver genes. And so those lentiviruses are then expressing, using that gene payload, either AGAT, GAMT by themselves, or both together. Is that and the empty vector is basically just, it is filled with an RNA genome that does not encode either AGAT or GAMT. It's just, we use that as a control because we'd like to see that any phenotype that we observe is not caused by having the viral infect the cell, but rather it's due to the specific effect of delivering AGAT and GAMT.
That's great. Thank you.
Okay.
Yeah, and so like Steve said, when we overexpress this AGAT and GAMT, we overexpress them in HEC293 T cells, and then we went and measured the creatine in the cytoplasm. And so as you can see with these two tall bars, AGAT and GAMT, and then AGAT V5 and GAMT V5 with that tag is significantly higher compared to the empty vector.
Perfect. Yeah. And so if you actually line up the data and you do a quantification, so this over here, this empty column would be how much creatine a HEC293 T cells normally has inside of its cytoplasm. And it's about 25 nanomoles of creatine per mg of protein. So suffice it to say, let's take that as the baseline for the cell, is that it would have about 25. When we overexpress AGAT and GAMT, we can increase that concentration on average to about 200 to about 230 or 240. So that's something like an 8 to 10-fold increase in the amount of creatine inside that cell. And I think that's important because inside of a neuron, remember, what we're trying to achieve is millimolar concentration. Here, this isn't given the concentration in millimolar. So that's in order to extrapolate that data, you have to measure the volume of the cytoplasm, which we're working on a technique now to do accurately. But this is a standard technique to quantify creatine inside of a cell, which is just to quantify that creatine per the amount of protein in each cell. And suffice it to say, we can boost the creatine inside of a HEC cell when we deliver AGAT plus GAMT by about 8 to 10-fold. And so that's a very large improvement in creatine. And we are going to quantify what this means at the concentration level as well. Because again, our goal to get sort of a situation that represents what would be present inside of a healthy brain, it has to be probably in the millimolar range in order for it to be to function. And so it's important that we can do this on a large scale, and we have to see if this approach is getting us there. But suffice it to say, it's much higher than the natural level of creatine inside of a HEC cell, which is good news. And so we wanted to see what the AGAT and GAMT expression look like inside these patient fibroblasts. And so Chloe did some immunofluorescence, and she's really become an expert at immunofluorescence very quickly. And she stained some, these are the patient fibroblasts, which have that same mutation for AGAT and GAMT. And so do you want to kind of describe this experiment?
Yeah, so like Steve said, these are the patient fibroblasts. They have a loss of function mutation in the creatine transporter. And so this is more immunofluorescence data. It's just another way to show our overexpression of AGAT and GAMT via those lentivirus delivery mechanisms. So in the top row, you're seeing the cells that we just delivered an empty vector to. So these vectors did not have AGAT and GAMT. And then in the bottom, you can see that overexpression that we achieved with the lentivirus. And something about these, we introduced the lentivirus and we selected with blastocidin for the cells that had received it, and then we passaged these many times. So even after multiple passages, the cells were able to retain those proteins and stably overexpress them over a long period of time.
Yeah, so that's great. And so what she's done is, thank you, Chloe, is she's highlighted here, each blue dot is a different cell. And it shows up, this is a stain for the nucleus of a cell, which is the center part of the cell, which contains the DNA. And so you can see each of these blue dots is a different fibroblast. And within this field of fibroblasts, right, you can see that there's very little to no staining for AGAT. There's a little bit of staining for GAMT here. And when we overlay all of those, you can see these fibroblasts. And it's important that these were delivered to empty vector control. So this is what a fibroblast would look like that we didn't deliver AGAT and GAMT to. And then when we use our gene therapy approach to deliver AGAT and GAMT, you can see that there's much, much higher expression, just like on that Western blot where we could see those two bands that appeared when we delivered them. Here we're delivering both genes together, and you get a lot of AGAT expression and a lot of GAMT expression. And because we're not forcing the cells to express AGAT and GAMT, over many passages, if it was harmful for the cells to do this, then what usually happens is the cells that don't express a lot of protein sort of win out. And so they will outgrow their neighboring cells that express a lot of AGAT and GAMT. So kind of the first indication that this approach might be safe or well tolerated was that we could grow these cells over many passages, which is literally where we just take the cells from one dish, dilute them a little bit so they don't overgrow, and move them into a new dish. Over many passages, these cells retain their expression of AGAT and GAMT, suggesting that at the very least, this isn't harmful and it might actually be beneficial for these patient fibroblasts. And so when we zoom in on these cells, so now not looking at a field of hundreds of cells, but looking at maybe a handful of cells. Here, these blue dots are each the nucleus for a different fibroblast. And you can see in the empty vector control, we don't really detect anything above background for the AGAT empty vector infected cells. And for the GAMT, it's very hard to appreciate from this image. There's not a lot of GAMT again, that band on the Western blot is also very, very small. But when we deliver AGAT and GAMT, you can see they're overexpressed at very high levels. And the AGAT looks like these little strings inside the cytoplasm. So here's the nucleus, that blue dot, right? And AGAT is normally targeted to the mitochondria. And so the mitochondria are an organelle that's inside of each cell, and they're sort of the powerhouse of the cell. So they generate energy for the cell. And we've known for a long time that AGAT has a mitochondrial targeting sequence. So the AGAT protein is supposed to go to the mitochondria. And there was some really great work out of Andreas Scholze's lab that showed that, in fact, the AGAT N terminus is what leads it to be targeted to the mitochondria. And that's what we've seen by co-localization of mitochondrial markers by immunofluorescence. And here we see it. that those green strings are mitochondria that contain AGAT that we're overexpressing. And the GAMT, we know, fills the cytoplasm and a little bit inside the nucleus. And that's exactly what we see here. So the pattern of expression, one, it's high level expression, which is good, and the cells are tolerating it. And two, the pattern matches what would be expected for these two proteins and where they should be going inside of the cell. And remember that these are patient fibroblasts. And so here's just a three-dimensional image where we've taken a stack of Z images, so making thin slices through the cell. We stitch them all together, and then again, we make them rotate in three-dimensional space so you can see the entirety of the cell at once. And just showing you the string-like organelles that are inside the cytoplasm expressing AGAT, which are the mitochondria, and the expression of GAMP throughout the cytoplasm. And then you can see the shiny blue nucleus inside of each cell. And you don't see this when cells are delivered the empty vector control, which is exactly what we'd expect. And so then the question is, okay, they seem to hold on to these genes when they're overexpressing them. How does their growth rate look? How does their survival look? And so one way that we can do this is we can take these fibroblasts that have this mutation that are overexpressing AGAT and GAMT or the empty vector, and we can quantify how well they grow inside of a cell culture dish. And for this, Chloe put them inside of an instrument called an IncuSite. And an IncuSite is just a fancy camera. All it does is it takes that little plastic dish that the cells are growing in, and it comes to those cells every 6 hours and takes a photo. Comes back in 6 hours and takes a photo. And then using that image, what it can do is it can count over a course of 3 days. All right, well, every 6 hours I've taken a picture. How many cells are there after 6 hours, after 12 hours, after 18 hours? And so you can literally watch the cells grow almost in pseudo time, right? Over the course of 3 days. And when we do that, so Claude, do you want to describe this?
So we did this a couple of times. We chose to supplement media with creatine precursors. So these are just the amino acids that are used to produce creatine naturally. And then we tried it without creatine precursor supplement. We didn't really find a significant difference with that, but with all of our experiments, the cells that received AGAT and GAMT grew faster and became more confluent quickly than the cells that received the empty vector. And so it would be helpful alone to know that our gene therapy plan didn't harm the cells, but it's extra helpful to know that it might help them to grow faster or be more healthy.
Yeah, perfect. Yeah. And so we are cautiously optimistic that this is maybe not, certainly not harmful, but maybe even beneficial for certain so at least for fibroblasts that are deficient in CT1. And so we want to try this again to be sure of that in multiple different patient fibroblasts. And again, this is where it's really helpful to have a community of families and patients who are willing to donate their fibroblast samples so that we can try this across a diversity of mutation types and a diversity of genetic backgrounds. And so at least in this first try, what we were able to see is that cells that are stably expressing AGAT and GAMT at very high levels seem to proliferate acceptably in culture, and even better than cells that got the empty vector control. And so the next thing that we turned to was we have a line of wild type fibroblasts. So we did this with the patient fibroblasts, and we're currently characterizing these wild type fibroblasts. But suffice it to say, just like in the patient fibroblasts, we can overexpress AGAT and GAMT, and we can detect very high levels of expression when we deliver AGAT and GAMT by a lentivirus to these wild type fibroblasts. And so these are some early preliminary data from those experiments. And so Chloe, again, has performed immunofluorescence on the wild type fibroblasts, and she sees high levels of AGAT and GAMT expression here by immunofluorescence, where each of these blue dots marks a different cell. And what's interesting to us, oh yeah.
Steve, I want to do a quick section summary before we keep going, and I think we have a question also in the Facebook live feed that we can get to. But as a quick sort of section summary to make sure that I'm following along properly, our hypothesis was to see whether you can exogenously add AGAT and GAMT to human cells,
Yes.
and see if they will function. So we first tried this using a lentiviral vector, which is basically a way to get these proteins into the cell. We tried this in hex cells.
Yep.
And then in the hex cells, we saw by one technology, so Western blot, that you are able to see these exogenously added proteins being expressed. Then we tested the same thing in CTD patient fibroblasts. So there also we were able to see that the protein is expressed by or protein and enzyme being the same thing. It is expressed by Western blot, and the confocal imaging also shows us exactly which parts of the cell these two proteins are available in. In the hex cells, we were also able to see that you do have creatine being produced by these exogenously added enzymes. Okay.
Perfect. Yep.
Making sure I'm on the same page. All right. And then I'll quickly hand it over to Heidi to tell us what the questions are that we've received so far.
Great. Yeah.
Sure.
Thanks.
Oh, sorry. First question is, could you get too much growth or growth be too fast, causing issues?
Yeah, that's a really great question. And so a lot of this, I mean, is early work that we're doing in cell culture. And I think ultimately the long-term goal is to try this out in vivo. And in vivo, the circumstances are quite a bit different. So when you take a fibroblast out of a body, and you put it on a culture plate, that is if you can imagine, it's a cell that was normally surrounded by the rest of its friends, and it's what's called the extracellular matrix inside of your own body. And when it's growing on a culture plate, it actually simulates for that fibroblast what it would be like to be growing inside of a wound or something where it's not surrounded by all these comforting signals that it doesn't need to proliferate and grow. And in fact, when you cut your arm or you cut your leg or you get in an accident, what happens is you have a gap in the tissue, and the fibroblast will move in. And this is actually what causes a scar to form over if the cut is large enough, is that those fibroblasts proliferate and they grow and they synthesize collagen, which fills in that hole, right? And so that's a lot like what's going on inside the cell culture plate. And so when we monitor the growth of the cells inside the culture plate, it's almost like an indirect measurement on how healthy and able they are to grow, but we don't expect to see that inside of the body. So inside of the body, the cells that we're actually delivering AGAT and GAMP to would be surrounded by those extracellular cues that everything is okay. And so the fibroblasts, for example, that are inside your body aren't ultimately the target cell that we're hoping to deliver to, but instead we're hoping to deliver this gene therapy to cells inside of the brain. But suffice it to say that if we had targeted cells like fibroblasts inside of your body, our hope is that they wouldn't be proliferating, is that it is a nice metric that they can proliferate if called upon, right? And that's what we're seeing in this plastic dish. But inside the body, they wouldn't be receiving those same cues telling them, hey, you need to grow and fill in this hole that's inside the body. Yeah. So
Hey,
it's a bit of an artificial system. Yeah. If that makes sense.
That makes perfect sense. And that leads very nicely into the second question, which is, are you planning to do similar experiments in differentiated cells?
Yes, very much so. So actually, if you'd like, that's a very prescient question, because that's the next segment of the talk, and really the work that John is pioneering. So suffice it to say, we can do the same thing in wild type fibroblasts, and we see the expression as well. And again, Chloe has done some really nice immunofluorescence imaging to show that that works. But then the next thing that we're doing is we'd like to try this not only in primary cells, but in differentiated cells that are of human origin. And so in order for us to do that work, what we need a human cell type that we can convert into almost any cell type that we want. And in order, luckily for us, Shinya Yamanaka has worked out the necessary genes that are required to convert a human cell into what's called an induced pluripotent stem cell. And that is called iPSCs. So what these are these were these are wild types. So these are from a healthy control, where they took fibroblasts and they overexpressed a set of genes that converted them back into something that's almost like an embryonic stem cell. It's almost like a cell that can become any other cell type. And when you do this, these iPSC cells will grow in culture, and then you can induce them to differentiate into mature cell types. And that's the next phase of this project. And luckily for us, again, we have amazing patient families and patients who have donated their fibroblasts to the Coriell Institute. And the Coriell Institute has converted those fibroblasts into these iPSC cells. So for us, that's a very precious resource. And so the first thing that we're doing is we're going to try to make healthy human neurons from iPSCs to test out the differentiation strategy. But then our immediate next step is to do this with patient iPSCs and to differentiate them into human neurons that we'd have in a cell culture dish to deliver AGAT and GAMP2 and see if that causes them to produce creatine and how much. And so for this work, John has been culturing these iPSC cells. And what we're just showing you here is that he's become really, really an expert at getting them to grow inside culture. And they make these nice, happy colonies. And we can deliver genes to them. And so this was our very first approach at this. So what we did is we delivered a gene that is from a jellyfish. And so that jellyfish protein, suffice it to say, we're not interested in using it for gene therapy. The only thing we use it for is that it glows green. So it's called the green fluorescent protein, and it was taken from a jellyfish, and you shine blue light on it and it shows up green. And so we can use that to test whether or not we can deliver genes to these iPSC cells. And so here's just a light microscope image of the iPSC cells growing in culture. And then we deliver to them the GFP protein, and we see it being expressed inside these iPSC cells. And so our next step is to essentially build this system out in order to deliver a gene called neurogenin-2. And neurogenin-2 is a protein that tells the cell, hey, I know you're an iPSC cell right now, but I want you to be a neuron. And so we're planning to make a system that will deliver neurogenin-2 to these iPSC cells, both from healthy control iPSCs and in patient iPSCs, and then turn them into neurons, and then do the next step, which is deliver AGAT and GAMP. And so John I don't know if you want to summarize the workflow, but it's essentially that.
Yeah, that's essentially it. And once we give them neurogenin-2 and we have a stable line of these iPSCs, we can cryopreserve them so that we can repeat the experiment over and over. And then once we have that stable line, we can introduce docs, which will tell them, hey, you're an iPSC right now. This will tell them to become a neuron, essentially. So once we induce with docs, it'll tell them to become a neuron. We'll grow a whole plate of neurons, and then we can deliver our AGAT and GAMP, and then we can measure the creatine within neurons.
Perfect. Yeah, thanks, John. So a great question, and that's definitely our next step. Is we have these fibroblasts which are actively growing inside of culture, and in a way they are a differentiated cell, but we really want to see this in a terminally differentiated post-mitotic neuron, and I think that's the direction we're heading to, and yeah, awesome question.
That is all the questions that I've received so far.
Okay.
It's pretty neat.
Yeah,
Very exciting stuff. I was, wondering if you could. So obviously, I understand that we did this in Hexels, we did this in iPSCs, and now we're going to do, or we did this in Hexels, we did this in fibroblasts, and we're doing it in iPSCs. So can you just on at a high level tell us about the significance of like why is it important to test this type of hypothesis in all of these different cell types? Which one's going to be the most relevant
yeah.
for the disease that we're studying and why?
Totally. Yeah. And so the endogenous concentration of creatine inside of a fibroblast is really low. And I think Dr. Mabunza's talk at the ACD forum this year, they actually measured the endogenous concentration of creatine inside of a fibroblast, and it was in like the single digit micromolar range. And so micromolar creatine concentration is probably sufficient enough for a fibroblast to function. But again, remember, it all comes back to that concentration thing where inside the brain, we probably need to achieve millimolar concentration and 10 to 20 millimolar concentration to see an appreciable benefit. And so different cell types have different amounts of creatine inside their cytoplasm. For example, inside the skeletal muscle, it's very similar to the brain, where you have on the order of tens of millimolar of creatine inside the skeletal muscle, and the heart is very similar. And so I think using fibroblasts and HEC293 T cells are a useful model to screen for whether or not we can deliver the gene, whether the proteins with or without the tag function. And so far, what we've seen is that those work in every context we've tried. But ultimately, we need to do this inside of a cell type and to quantify the amount of creatine that we're making inside the cell type that really matters. And for our hypothesis is that cell type is the neuron. And again, it comes back to the fact that neurons express CKMT1, so that creatine kinase. And in order for a cell to use creatine, it must have a creatine kinase. And without getting too much into the weeds, the mitochondrial version of creatine kinase is usually expressed in cells that really rely on creatine, like the skeletal muscle and the heart. And that's why we're strongly suspicious, because the CKMT, that MT stands for mitochondrial, is explicitly expressed inside of neurons, or I should say exclusively. So that enzyme is very highly expressed inside of neurons. And so we think that those neurons are the cell type that need creatine inside of the brain. And so that's why we have to use those as our model system to test this approach to therapy. And so that's certainly the place we're going to. And it's useful to have the fibroblasts in Hexel data, but having patient iPSCs that we can differentiate into neurons, and then of course having the mouse tissue that we can use to collect and differentiate neurons from a mouse source are such vital resources for where we're going.
Thank you. Thanks, Steve, for elaborating on that. That's useful to hear and appreciate. I got to say, from thinking last year about this as an idea to try and thinking about how much progress we've made in just like, I know we got CDRC started only earlier this year, so you've been working on the project only for a few months. But even so, from an idea on paper to actually expressing AGAT and GAMT and actually seeing creatine being produced in not just one but multiple cell types, this is amazing. I'm really excited about what next year is going to bring in terms of validating some of these findings and testing it really further in a more complex system like a mouse, right? A mouse model of CTD, as well as findings in perhaps cell types that are obtained, relevant cell types that are obtained from a mouse model of CTD as well. So really, really exciting work and phenomenal progress overall. And your proposal has been a really great example of like thinking a little outside the box and moving the field of CCDS research ahead. And it aligns really well with what the ACD has been interested in doing. We are thinking about expanding our efforts further within CTD as well as to GAMT and AGAT deficiencies. And I'm positive that we will get there because we have an amazing community of supporters. Isn't that right, Heidi?
Yes, thank you. Thank you very much. Thank you, Steve and Chloe and John and Sangeetha and everyone for participating and posting your questions today. And it really is exciting to see these positive results so soon and exciting to really be a part of understanding your plan going forward and how you're going to validate this. And of course, also, I appreciated you pointing out that the safety concerns are always in the front of your mind, and that is why this does take some time, but I'm really excited about your positive results so far. And so I think I will wrap this meeting up by just again acknowledging that this project is part of the project funded by the ACD community. And so thank you to everyone who donated last year to our Holiday Heroes fundraiser. That is why this is all possible, quite literally. And we want to share, looking forward to 2024, some of our goals as we launch Holiday Heroes in just over a week from now. We are raising our goal to 350,000 this year because we would like to support quite a few initiatives. One and our biggest is the Creatine Deficiency Research Center Year 2, so we can continue this amazing work. We have fellowship projects that we are reviewing right now and will be announcing at the beginning of the year. And those are always very valuable to bring in new folks like Dr. Baker with big ideas and lots of energy to initiate important projects such as this. And we have a drug repurposing research project that comes from one of those fellowship programs and is growing into something bigger and exciting as well. We're also partnering with Dr. Laura Baroncelli and Michaela Fagiolini at Boston Children's Hospital in a project there. And we are having a conference next summer. So we have a lot on the horizon. And on that note, I wanted to share... I can't advance my slide. There we go. I wanted to share that if you would like to get involved and help in our efforts, you can visit creatineinfo.org/holiday-heroes, and you can get signed up to be one of our families and supporters of any kind that are hosting a campaign to contribute to the larger goal. And you can also reach out anytime to myself, to Emily at creatineinfo.org. We're all here to help you. We have lots of pre-formulated outreach materials, and we'll take cute pictures of your family and your kids and make you some custom graphics. And we want to support you and help you to help us to help these researchers to be successful. So thank you, everyone, for participating today, and we will post this recording in the next few days so you can go back and re-listen to everything. Thank you again, Steve. You always share so eloquently your research, and I learn more and more every time I listen to you. So thank you very, very much to you and your team.